Good morning students, welcome to LEELA'S tutorial. So today we'll know about molecular markers, types and their characteristics. So what do you mean by molecular markers? These are small molecules contained within a sample taken from an organism. So why you need to take the sample from an organism? What do you mean by the sample here? Sample is nothing but the DNA sample. It can be from the blood of that particular organism or from the hair root or the nails or any other sample containing the DNA such as semen, etc. Right? So here you are going to collect the sample from an organism so that you can know the characteristics of that particular source. Right? So here the molecular markers are mainly used in molecular biology and biotechnology in order to know the particular sequence of DNA within a complete pool of DNA. If you have many number of DNA samples from that particular all the pool of DNA sample, you can know the particular sequence present in that. Right? The particular sequence of DNA which is required can be known. So here if you see what are the different types of markers, basically you have DNA molecular markers and protein molecular markers. So where these molecular markers are mostly used in case of diagnosis, right? In case of diagnosis or in case of differentiating the DNA from the DNA pools, right? So here you have DNA molecular markers. They contain the information about the genetic disorders. That means here the genetic disorders are caused due to some variations in the genes, right? So due to these variation in the gene, when you know the genetic analysis of a person, if there is any defect in that particular sequence or in a particular segment of DNA, then that segment can be corrected by using your gene therapy, right? So here these markers help us to identify that particular genetic disorder or the location of the gene where the disorder is caused due to. Then you have the evolutionary history. You can know what is the relation between the different types of animals or the traits of animals or the breeds of animals, right? You can know what is the variations in their genome from the evolution to the complete history of life. Then coming to the protein molecular markers, these are widely used in the diagnostic tests like such as complex neurodegenerative diseases, that is mainly for example Alzheimer's disease. So in such diseases these markers are used for diagnosing, right? So if you come to the types of molecular markers, they are broadly classified into three types. First generation molecular markers, second generation molecular markers and third generation or new generation molecular markers. So here all these markers, they are having different names for each marker. All these are the examples of the markers, but you are referring them as first generation, second generation, third generation. That is based on their characteristics and based on the functions of these markers, they have been differentiated, right? And they have many modified forms also. So if you come to the first generation markers, example RFLP, RAPD and their modifications are used. What do you mean by RFLP? Restriction fragment length polymorphism, right? So here the restricted fragments are used in order to identify the particular sequence of DNA. All these we will discuss in detail in the next class, right? So next you have RAPD. RAPD is random amplified polymorphic DNAs. Okay, then you have the second generation markers. The examples are SSR and AFLPs, right? And then modifications. SSR is simple sequence repeats. AFLPs are amplified fragment length polymorphism. Then the new generation or the third generation molecular markers, you have ESTs, that is extension sequence tags, and SNPs, single nucleotide polymorphism. So all these are fragments of DNA either having the restriction digestion or rapidly amplified particular sites, or the single nucleotide is used to identify that particular DNA sequences. So we'll see all these techniques in detail, okay, in next class. Then what is the use of these molecular markers? What are the characteristics of these molecular markers? The first one is they allow amplification of particular sequence within a genome and comparison. So for comparison and analysis, you can completely know the genome of an organism. When you know complete genome of an organism, it will be easy for you to compare and analyze any genetic disease or anything. Second one is identify an abundance in genetic linkage between identifiable locations within a chromosome, right? So when you have the genetic linkage, right, when there is some linked gene, so within the chromosome, if you want to know the particular linkage, where is the location of the linkage, you can identify that. Then third one, identify small changes in mapping population, enabling distinction between specific traits. So what I mean by mapping? Mapping is nothing but the distance between the successive genes or the complete set of genes. So genome is nothing but a complete set of genes. So in that arrangement, the position of the gene and the distance between one gene to the other gene or the complete genes, everything, complete data will be present in the gene mapping. So once you know the gene mapping, you can distinguish the traits at which gene or which position of the gene these traits are differing, right? Then in the next use, it is particularly identifying the location on a chromosome to create a physical map. If you want to create a physical map over the chromosome, you want to identify that particular location. Again, by using these molecular markers, you can identify, you can prepare specific sequence as a molecular marker, and you can identify that particular location or position over that marker. Then last one is identify the alleles of an organism of particular trait. So here you can identify whether the gene or the allele is present in duplicates or in many number, right? Mostly the alleles are biallelic, or you can identify any polyallelic forms are present in these markers or not. So here the molecular markers play a very important role in identifying the one person from a pool of population in criminal cases and all, right? So we'll see all those techniques, how they are used in detecting these in the next class. If you have any doubts, put it in comments and subscribe.